And with that, I'd like to introduce Susan Gilliland again to introduce our program for tonight. Susan?
Thank you, Ron. Well, it's our pleasure to have Dr. Keeney and Dr. Billerman with us from Cornell Lab of Ornithology tonight. Dr. Brooke Keeney was the managing editor of Birds of North America and Neotropical Birds Online, and now the Birds of the World at the Cornell Lab. Brooke handles the publication of species accounts from the draft to final product, a rather daunting task for 10,824 species. Some of you may remember Brooke from bumping into her birding when she lived in the South Bay area of LA County. Prior to Cornell, Brooke worked as a science editor and content developer for Pearson Education and taught at Northeastern University. She's also worked as a wildlife biologist in Hawaii and the Channel Islands and volunteered as a wildlife rehabber, specializing in owls and other raptors. She earned her undergraduate degree at UC Berkeley, where she worked as a curatorial assistant, and her graduate degree at UC Riverside in ecology, evolution, and organismal biology. Dr. Shawn Billerman is a science editor with Birds of the World, where he has a special interest in bird families and taxonomy. As science editor, Shawn helps to edit and revise accounts that come in from authors around the world. Prior to working with Birds of the World, Shawn was a postdoc at the Lab of Ornithology, where he studied hybridization across different hybrid zones from the Great Plains of North America, including indigo and lazuli buntings, rose-breasted and black-headed grosbeaks, eastern and spotted towhees, and Baltimore and Bullock's orioles. He used genomic data from specimens in the Cornell University Museum of Vertebrates, as well as climate models to understand the important factors that have shaped these co-occurring hybrid zones. Shawn earned his undergraduate degree at Cornell University and his graduate degree at the University of Wyoming, where he studied a hybrid zone between red-naped and red-breasted sapsuckers. Tonight, Dr. Keeney and Billerman will introduce us to Birds of the World, a comprehensive resource that provides in-depth information on all 10,824 species and 249 families of birds. It's an immense project that includes partnerships with ornithological groups and a network of experts from around the world. Brooke and Shawn will help us to understand how Birds of the World keeps up with the ever-expanding research that is published on birds and what goes into maintaining the taxonomy that underlies it all. So please welcome Dr. Keeney and Dr. Billerman.
Thank you all. It is so nice to be here. Let me share my screen. Yeah, so thank you again for having me, and thank you for that introduction, Susan. And it's really nice to see some familiar faces. Like Susan said, I used to live in the South Bay, so it's really nice to see this community of people. And knowing a little bit about this group and getting to meet you all in the field and birding, I know that there is a lot of insight and expertise on this call. So in many ways, this group is the sort of audience that we're most hoping to engage with Birds of the World. A savvy audience that's deeply invested in birds at both the local and the global scale. So as such, I want to stress that Birds of the World is really your resource, and we value your input in how the site evolves. So as we go along, feel free to jump in with questions or with comments. It's great for us to hear what you think Birds of the World does well, and it's even better for us to hear where you think we could improve or what we could optimize or any other insights you might have. So to start tonight off, I'm going to talk a little bit about how Birds of the World came to be and what we have available on the site today. And then I'll explain some of our current challenges, which will culminate in Shawn, who will be discussing one of our greatest challenges, which is the science and the process of taxonomic updates for more than 10,000 species accounts. So for anyone who is less familiar with the site, Birds of the World is a scholarly, comprehensive natural history resource for the complete collected biology of every bird in the world. This resource combines species accounts written by a global community of experts with millions of observations from eBird, cutting-edge data products from eBird Science, and multimedia from Macaulay Library. Birds of the World itself is a relatively new project, having launched only in March of 2020. However, the foundations of Birds of the World go back more than 30 years. The content in Birds of the World is derived from 4 different source projects: Birds of North America, or BNA, Neotropical Birds, or NB, Bird Families of the World, and the Handbook of Birds of the World, or HBW. So to give you a little bit of history of the projects, both BNA and HBW were founded in 1992 rather, as print publications. BNA was originally helmed by the National Academy of Sciences and the AOS, which was then the AOU, and HBW was helmed by Lynx Editions. So although both projects had similar aims, they each had different foci and strengths. HBW excelled in its broad coverage, offering a snapshot of the essential biology of every species of bird, while BNA became the gold standard in deep, authoritative species accounts for a geographically limited set of species. So as both projects matured, it became evident that print publishing was not going to be agile enough to keep up with the speed of science. And both projects made the transition to a digital environment along becoming BNA Online and HBW Alive. And so with that transition, Cornell took over stewardship of BNA, and alongside it started the Neotropical Birds, or NB project, thus increasing its coverage to more than 1,500 species accounts authored by 2,000 authors. So with the transition to digital, all 3 projects began to explore ways to further enrich their resources. BNA and NB began to incorporate the richness of eBird data as well as media from Macaulay Library, while HBW developed the Internet Bird Collection, which was a collection of captioned images that was interwoven into the species accounts, and it was also a standalone repository for bird media. In 2015, Cornell and Lynx published The Bird Families of the World, which was written by David Winkler and our own Shawn Billerman and Irby Levitt. And this was the best available single volume entry to avian diversity. And it was also the first formal collaboration between Lynx and the Cornell Lab. And that sort of paved the way for the eventual merger of all 4 of those projects. So what you'll find on the Birds of the World site today is still very much a product of the merger of those source projects. At minimum, there are basic species accounts for every one of the 10,824 species currently recognized by our taxonomy, which are accompanied whenever possible by gorgeous illustrations, rich media, and a variety of range maps. So you can take a look at our most recent updates on the homepage, which is shown here, or you can use the Taxonomy Explorer to drill down by taxonomy or by location. You can also use the Species Explorer or even the Surprise Me feature, which can be a fun way to kind of learn something new or explore the endless avian diversity. And there are a variety of other features on the site that don't relate directly to species accounts that I won't be talking about today. These include our blog, the Key to Scientific Names by James Jobling, as well as HBW's ornithological notes. So all of those bear mentioning, and you can explore them in greater detail on the site itself. So in addition to species accounts, there are also family accounts for all 249 families of birds, which contain text, maps, stats, and the ability to explore and visualize diversity across genera within a family, as well as within a larger phylogenetic framework. So shown here is a typical species account introduction page. As you can see on the lower left, each species account is subdivided into different aspects of the species biology, so you can easily get to what you're looking for. And it's accompanied by expert curated media, which includes, a really stunning, large, charismatic image, which you can see front and center there. And then an essential set of images that you can see the scroll for on the top right, which gives you really the fundamentals of what you need to know about that species appearance, its behavior, its association with the habitat, anything else that can be easily shown visually, including audio and media. Or video. So you can also get a review of the common names for the species, which is shown as a clickable pop-up in the blue underneath the scientific name, as well as the species entry into the job link key to scientific names, which is denoted by the blue little I next to the scientific name. So each account also has a revision history where one can see and explore each of the source project's unique iterations of that account. And I really like this because it allows you to sort of understand that each one of these species accounts is really a living document, and there's a history that goes into each one of them that's important and unique on its own. So I mentioned that I'll be talking about some of our challenges, and the first of our major challenges is really to expand upon a basic sort of coverage for every species. And of course, logically, when we consider what's possible for a given species, there are some species that have really extensive publication records and some for which very little is known at all. And of course, all the different shades in between that. So if you take a look at some of the species we've recently published, there's a great representative sample of the different levels of revisions that are possible. On the deeper end of the spectrum, there are full BNA style revisions that were written by returning authors. Some recent updates in this category include the accounts that I've highlighted, the yellow-breasted chet, the black vulture, the white-eyed vireo accounts. All three of these are really exemplary accounts by authors who are updating the work that they originally did for BNA. And each is a fantastic example of how rich and valuable an account can become over time. The authors are really taking the strong foundation that they originally wrote for BNA and building upon it and making it richer, more engaging, and more interwoven with eBird and with distribution maps that show dynamic aspects of its range and its habits and its behavior that really wasn't possible before. So there are also some incredibly deep accounts by first-time authors. And today I'd like to highlight both the Eurasian griffon and the European bee-eater accounts. So I know the text on this is small. However, I hope that you can take the time to explore these accounts in greater detail later. These accounts were written by authors who became contributors since the advent of Birds of the World. And both of these accounts are incredibly detailed and rich, and really a testament to the dedication of authors from around the world who are willing to put in significant work to make this resource what it is. So both of these accounts are incredibly thorough. The Bee Eater has more than 700 references, which Shawn can attest to because he edited it. So most of them are new additions. I want to highlight these accounts not just because I'm proud of their content, but because they show something really important about the value of this resource around the world. Birds of the World really is the singular project for scholarly digital natural history, and it's really exciting to see the global community respond and grow and engage with the project. So going back to our recent updates, I also want to highlight some revisions for species for which there's just generally less known. These species here are all written by new authors, and they're just much less well-known relative to the other accounts that I showed you. So for something like Swinnerton's robin or Ochraceous piculet, these experts have done the most complete job possible for what's known about the species, and we find it equally important to highlight when information is not known for a species. So part of the utility of these species accounts is that they can be a really effective tool to highlight where further research is needed, and both of these excellent accounts master that balance. So we've been really incredibly lucky to work with such a motivated and scholarly and enthusiastic community of contributors thus far. However, the second of our major challenges is building that contributor community to ensure that it's diverse and representative of experts of all levels and stages. And we acknowledge that both BNA and NB were written from a geographically biased perspective, and we are most familiar with those communities. So to truly make this project the global effort that we envision, we need more help. So over the past 2 years, we've engaged in several collaborations or so-called content for access partnerships with global ornithological societies and NGOs. These include Birds Caribbean, Bird Count India, ROC out of Chile, SAVE Brazil, OSME, and others. So in these partnerships, a partner group agrees to keep a selection of species accounts up to date in exchange for free Birds of the World access for their constituency. These partnerships have enabled us to leverage the strengths and the focus of our partners to engage with contributors that have deep, authentic experience with their species. And it helps us to diversify our global author community. In return, we're able to offer our partners a service that they can use to build and incentivize their community. So thus far, it has absolutely been a win-win. Looking back at our recently published accounts, all of these revisions that are highlighted here have come from resulting of a partnership, which really gives you an idea of how vital and energized these collaborations can be. Not only are we publishing really exemplary content from them, but we get the benefit of a deep, regionally specific community. So that said, another challenge we have to deal with is how to respond to really exigent publication needs. So as Shawn will detail later, when we merged our 4 source projects, there was a slew of issues requiring our attention. And it can be really difficult to find ways to address these issues if we're only looking for contributors opportunistically. So most disruptive among these issues were accounts for which there was a taxonomic mismatch between HBW's treatment of a species and the treatment we follow, which is the Clemens checklist. So to help us address these sort of time-sensitive issues, we have a team of consultants or associate editors, some of whom are shown here, who are ready to lend their expertise to revise accounts. So starting at the top left, there's Harold Greeny, Pamela Rasmussen, Washington Wachira, Rosalvi Juarez, Natalia Garcia, Per Alstrom, Gerardo Marron, Narjila Mora, Wichinan Lim, Guy Kerwin, Philip Maleko, Steve Molodnov, and Yoav Perlman. There are others too. Each of these specialists really brings with them a wealth of expertise and a lot of ornithological strengths. And through their efforts, we're able to thoughtfully revise the content that needs it the most. But these teams are also shaping the content we're putting out there in important ways. So for example, Peter Pyle, who's shown in the center here, who's been a longtime contributor to BNA and now Birds of the World, is working with an international team of experts on the molts and plumages sections of accounts. So this team includes Josef Kiat, Twenchom Hansasuda, and Arnau Bonan Barfel, among others. And they're really leading the charge in formalizing the use of digital images from Macaulay Library to determine avian molt strategies. In doing so, they are amassing an incredibly useful and centralized catalog of curated and captioned images that show details of molt. To my knowledge, nothing like this has been attempted at this scale before. But even with all of these updates and all of this talent, the challenge of keeping species accounts for every bird in the world up to date is a really significant one. So the continued success of this project is directly related to contributors and users and birders and engaged, thoughtful individuals like you all. So I hope that what I've shown you today has excited you about this project and made you think a little bit about how you might get involved. So we aspire to be dynamic and a current resource, so we're always looking to update almost all species accounts to one degree or another. If you are interested in getting involved with the content or the media of species accounts, we're looking for qualified authors to lead full or partial revisions or to join revisions, or reviewers who are willing to help in the review process. I'm oversimplifying the authorship situation, but that's something that I would be happy to discuss with you on an individual level. So for context, a full revision would be one in which a reviser evaluates and attempts to update all sections of an account, whereas a partial revision would be an update to only one or two sections. So something like sounds or appearances or something that aligns with a specific area of expertise. So that can be a great targeted way to make an impactful contribution in a relatively short amount of time. So beyond formal contribution, our team is always looking for feedback, like I said. So we know that different audiences have different needs from our site. And so one area that I think overlaps well with the scope of this particular group is the need to develop content that can really help with complex field identification challenges. So in the next year, we're hoping to further develop our identification and similar species sections into something that can be really useful in the field, potentially customizable for the different layers of identification challenges that are possible. So if this is something that inspires you or interests you, please feel free to get in touch with me. So in summation, I hope that I've given you some background about what our site is and what you can find on it and the challenges involved in managing it. But that said, one of the biggest challenges that I've only just alluded to is the challenge associated with keeping species up to date when the species themselves are changing. So I'd like to turn it over to Shawn, who will talk in greater depth about how we handle taxonomy on a global scale.
Thank you so much, Brooke, and thank you to everyone for coming. I'm really happy to be here. Oops. Okay, that look okay to everyone? Okay, so as Brooke said, I am going to be diving into the wonderful world of taxonomy and classification. So in Birds of the World, like all other projects at the Cornell Lab of Ornithology, including eBird and Merlin and All About Birds, the taxonomy is supported by the Clements Checklist, which the Lab of Ornithology has maintained since 2007. Led by Tom Schulenberg and many other people who contribute, the checklist gets nearly annual updates. The last update in August of 2021 resulted in a list that now recognizes, as Brooke mentioned, 10,824 species and 249 families. And some of those newly recognized species include Pinau ant pitta, which I will get to in more detail later, the Kilambaro cysticula, which is a brand newly described species from Africa, and the short-billed gull, which many of you are probably familiar with. It was formerly known as mew gull and has been split from its relatives in Europe and Asia. But before really diving into the nitty-gritty of Birds of the World taxonomy, I first wanted to quickly review some of the basics of taxonomy and classification. So in taxonomy, we recognize all of these different categories, including species, families, orders, but what are these groups? So basically in biology, we need a way to communicate about the diversity of life. And in order to effectively communicate, there are some general rules to follow so that when we say something about a genus or a family or a species, everyone knows what we're talking about. Now, the most important rule of taxonomy and classification is that each named group that we talk about represents a monophyletic group. So to think about what a monophyletic group is, think of a group of species. If that group is monophyletic, it means that every descendant of a single ancestor is present in that group. If one descendant of an ancestor is excluded, it's no longer considered a monophyletic group. By the same token, if something is included that did not descend from that single common ancestor, then that group is also no longer monophyletic. This simple rule applies to all scales, from kingdom, phylum, class, all the way down to subspecies and everything in between. Even before people really understood what evolution was, and thus what monophyly was, the goals of classification have always been the same. In the Linnaean system for classification, which is more or less what we use today in a modified way, groups of organisms that looked more similar to each other and shared certain characteristics and traits were placed in the same group. So to really understand monophily a little bit more, let's take a look at this phylogeny. So a phylogeny is a way to visually represent how organisms are related to each other. In this fictional phylogeny I have on the screen, we have species A through J on the right, and the pattern of branching shows which species are more closely related to each other. So in this case, species A is most closely related to species B, species E most closely related to species F, and so on. And as you move from left to right across the screen, we're moving through time. So the left is the past and the right is the present, where we have all of our species. The point at which a phylogeny splits into two or more branches is a node. And that node represents the most recent common ancestor of all the branches that come from it. Or it's the last time that the species at the end shared an ancestor. All the lineages that share that common ancestor have some set of characteristics in common that can be used to classify them and understand how they've evolved. So I've mentioned that monophyletic groups are the key to taxonomy and classification. So if we look at these three boxes, all three of these boxes that are drawn represent monophyletic groups. Each contains an ancestor represented by the colored dot at the base here, and all of its descendants. These monophyletic groups represent the building blocks of taxonomy and classification. Every name that you see, whether it's a genus, a species, a family, or an order, represents a monophyletic group, meaning that every individual in that group is more closely related to each other than they are to any other individual in another named group. Now, let's take a look at this box. This box does not represent a monophyletic group. The most recent common ancestor of species H, I, and J was all the way back here. So that means that this group in the red box excludes all the other descendants of that single common ancestor, species A through species G. And that's important to think about for later examples that I'll get to. But for now, just remember that pretty much everything that we talk about in taxonomy and classification is based on monophyly for the most part. So I've already mentioned that some of the different levels of classification, including species, family, and order. And if we're talking about birds, we're talking about the class Aves. Every bird is in the class Aves. Now, ostensibly, most levels of classification don't really have an actual biological meaning. Class, order, family, and genus are merely human constructs to help us kind of communicate about different levels of evolution. Species are arguably the only level of classification that have biological meaning. For instance, a cardinal knows what other cardinals are and also knows that chickadees and sparrows are not cardinals. Cardinals represent a discrete population of interbreeding individuals that does not interbreed with another discrete population. This example makes it seem really easy to tell what a species is, and for the most part it is, but there are cases that are not easy and straightforward, and this has led to extensive debate over what species actually are. There have been dozens of proposed methods for delineating what a species is. In ornithology, there are two primary definitions that people use: the biological species concept, which generally speaking, all populations of a species are roughly defined as being able to interbreed with all other populations. And then the phylogenetic species concept, which focuses on evolutionary history of a group and classifies species as the smallest group of populations that are all descendant from a single common ancestor that have some distinguishing characteristic. So getting back to what I mentioned on the last slide, genera, families, and orders. These don't strictly have biological meaning. They're shorthand for collections of species at different levels. They're not really a thing unto themselves, but they're a way to summarize higher level patterns of evolution. They really help us to understand how diversities actually evolved. And they help us to understand how groups are related to each other, and they can help us to learn about the natural history of poorly known species. If we know what that poorly known species is related to, it may help to guide us into understanding a little bit about its natural history. And in some cases, understanding higher level diversity can also help us to establish conservation priorities. Understanding classification and how birds are related to each other can also tell us a lot about how different groups originated and some of the processes that may have contributed to their evolution. So in this example, we're looking at the Struthiataformes, the branch of the avian tree that shares a common ancestor with all other living birds. On the one hand, we have the flightless ratites, including ostriches, rheas, emus, cassowaries, and kiwis. And on the other hand, we have the flying tinamous. Now, not only do all of these birds belong to the same group, the Struthiatiformes, but the closest living relatives of the flying tinamous are actually kiwis, emus, and cassowaries. This is really important because it means that even though most of this group is now currently flightless, flightlessness must have evolved multiple times. And it suggests a fascinating pattern of dispersal and divergence among this ancient group of birds. All right, so now that I've gotten the little lesson on taxonomy out of the way, we can get back to birds of the world. So I mentioned earlier that there are many ways to classify species and genera and families and orders for that matter. Currently, there are 4 major checklists of birds. And you'll notice that if you look at all 4 of these lists, the Howard and Moore checklist, the IOC checklist, BirdLife International, and Clements, which we use, all of these checklists take a slightly different approach to recognizing species. But even with these different approaches, they're remarkably similar. And that's because the underlying rule of monophily is still applied. Where the checklists differ are cases that are truly gray areas, and there isn't necessarily a right answer. It's a matter of degrees, personal preference, and underlying philosophy in what constitutes a species, a family, or an order. So when Birds of the World launched in 2020, as Brooke mentioned, it merged content from Birds of North America, Neotropical Birds, the Handbook of Birds in the World Alive, and the Bird Families of the World. In the first 2 resources, those were both maintained by the Lab of Ornithology, and so they both use the same taxonomy. But HBW Alive used a different taxonomy, and so syncing these 3 resources proved very challenging. Our first major task for Birds of the World was to figure out how to align the Clements checklist taxonomy that we used with the HBW BirdLife International taxonomy. In many cases, Birds of the World ended up adopting the taxonomy of BirdLife International as the changes were ones that we had already been considering. In cases where there are still disagreements, we worked really hard to align any discrepancies to the proper taxonomic group. And this is where what we call the group concept came in. While the group concept is not a formal taxonomic unit, it helped us to organize and classify distinct subspecies or groups of subspecies. Importantly, these are field identifiable species or collections of subspecies. So in the case of the yellow warbler, 3 field identifiable groups include the northern yellow warbler, which includes almost all the subspecies that breed in the US and Canada, the mangrove yellow warbler, which includes many subspecies from Mexico, Central America, and North and South America, and are characterized by the male's rusty head, and the Galapagos yellow warbler, which is characterized by rusty cap in males. And so you can see for these 3 different groups, you can see all the different subspecies that are included in a single group, and where each individual subspecies is not necessarily identifiable, all of these subspecies together constitute a group. And so the group concept of eBird really has a couple of other really important features. And most importantly, many groups represent possible future splits of species, and are an important way of collecting data on these taxa. In order to align BirdLife taxonomy with the Clements checklist in instances of disagreement, we are able to directly match the BirdLife checklist species with its Clements checklist group counterpart. So this alignment with our group concept ensured that no information on any of these differently recognized species was lost. Information in species counts is still displayed for both taxonomic units under the group concept. Further, using these groups, users can still track their own observations and media using eBird and search observations down to the group level, so no data are lost in this alignment process. And one fairly clear-cut example of how this works, we have the Birds in the World taxonomy, which recognizes a single species, the black-throated tit, which is found from China south to Vietnam and west to the Himalayas. BirdLife International treats the species as 3 distinct species: black-throated tit on the top, red-headed tit in the middle, and gray-crowned tit on the bottom, all of which neatly align with the 3 groups that we currently use. This allows us to show all the relevant information for these groups So while these are likely all valid species, we've delayed making a decision until more research is done, as genetic studies have found that it's a much more complicated situation than it may appear on the surface. And not all the relevant taxa have been included in the necessary studies. We've taken a slower, slightly more conservative approach to taxonomy because we don't want to have to reverse our decision or alter our arrangement further when new research is released. So, and it may seem like having to make corrections later might not seem like a big deal. Instability in taxonomy can cause a lot of confusion. It can hinder communication about groups, and it can also negatively impact conservation efforts when there's constantly changing the taxa that we're discussing. A much more complicated situation, but one that still highlights the value of using the group concept, is illustrated by looking at the taxonomy of the common cicada bird. This is a widespread member of the family Campophagidae, or the cuckoo shrikes, and is found in eastern and northern Australia, New Guinea, the Solomon Islands, and southeastern Indonesia. And as currently recognized in birds in the world, it's comprised of 29 different subspecies. Based on its distribution and the extreme variability in its plumage and voice across its range, some of the plumage variability is illustrated here on the page. It almost certainly represents multiple species, and BirdLife International has already split the species into 9 different species. However, that split, which is based on plumage and vocal differences, conflicts with genetic data. And there have also been some disagreements among taxonomists on how to split this, even using voice and plumage. So for now, we present information on all these taxa under a single species, common cicada bird. But the information is broken up by group, which aligns with the splits that were made by BirdLife International. So this is another case where more data are urgently needed to untangle this very complex and complicated system. A little bit closer to home, the group concept has also helped us to reconcile differences in how we treat what has become a very controversial and hotly debated topic, the yellow-rumped warbler complex. While the differences I've touched on in the previous 2 slides represented differences in how quickly we adopt changes, some mismatches between the different checklists represent true differences of opinion in how to treat taxa. The yellow-rumped warbler complex is one of those and possibly represents one of the most contentious taxonomic situations in North America right now. Research of the hybrid zone has suggested that the hybrid zone between the Myrtle warbler and Audubon's warbler, at the 2 individuals at the top here, they form a hybrid zone, and research in that hybrid zone has suggested that there's little evidence for assortative mating, or there's little evidence for Myrtle warblers to mate with other Myrtle warblers, Audubon's warblers with other Audubon's warblers. And the mating strategy there seems a little bit more random. And hybrids, shown here, are often produced. However, despite this little evidence for assortative mating, there's also evidence for selection against hybrids, suggesting that there is post-mating reproductive isolation. Proponents of recognizing multiple species in this group point to this selection against hybrids, as well as genetic differentiation among the groups as evidence that they represent distinct species. On the flip side, proponents of maintaining them as a single species point out the evidence of little assortative mating within the hybrid zone as evidence that they are not good species, and just represent well-marked subspecies. Neither of these approaches is wrong, and it represents a gray area of taxonomy where the application of the rules is not really clear. So far I've talked about and highlighted how we've handled differences between different taxonomic classifications in birds in the world. And again, I want to really stress that despite these differences, there is a lot of agreement and overlap across all the global taxonomies. The areas where there are differences represent true gray areas. But to wrap up this discussion of birds in the world taxonomy, I wanted to touch on some really fun recent examples where research has turned our understanding of certain taxon or taxonomic groups on its head. In particular, I want to highlight the ant pittas. In South America, ant pittas are often a highly sought after target for many birders, and many, as the ones shown on this page show, have subtly beautiful plumages in bold patches of browns, grays, blacks, and white. They're one of my favorite groups. They're like little Nerf footballs on legs. And they're really hard to see, and they're skulking, and you can almost never get a good look at them. They're way more often heard, except perhaps where some have been accustomed to come in to eat earthworms, a practice popularized by Angel Paz in Ecuador. But not all of them have these really awesome patterns and plumage patches. Some of them look, well, very monochrome and uniform, and not on the face exciting. But these somewhat drab muted plumages belie some incredible diversity. You'd be hard pressed to find consistent differences across the 6 pictures I have up. And for a long time, they were all considered only 1 or 2 species. Before 1987, all of these were considered Rufous ant pitta. Later, a second species, chestnut ant pitta, was recognized. Now, however, after a couple of recent studies published in 2020, the most recent taxonomic update in 2021 that came out recognizes 16 species in this complex. These extensive changes were based on genetic work that helped to guide extensive and detailed analyses of vocal differences. And in this case, extensive genetic sampling across the range of what was formerly Rufous and chestnut ant pittas, as well as some close relatives, revealed that neither Rufous ant pitta nor chestnut ant pitta were monophyletic, the golden rule of taxonomy. Further investigation also found that where there were deep patterns of genetic divergence, there are also vocal differences, which is really important in this group. Ant pittas are subausine passerines, which have innate songs compared to most of the songbirds that we have in North America, which learn their songs. The fact that these vocalizations are innate means that they are important indicators of species level differences. These species level splits were so cryptic in this group of ant pittas that many of the new species were not even recognized as subspecies, and thus required entirely new names. This major overhaul of ant pitta taxonomy also required an overhaul of ant pittas in Birds of the World. Led by Harold Greene, Andrew Spencer, and Peter Bozeman, Birds of the World has gone all in on updating all the ant pitta accounts, inspired by the need to create so many brand new accounts for this group of cryptic species. This effort by Harold, a leading expert on all things ant pitta, and Andrew and Peter, 2 experts in the field of avian vocalizations, has been a really exciting way for us to update the species accounts for an entire family in a systematic way. Guy Kerwin has also used this opportunity to develop a model for how we show the systematics and classification history of species and subspecies, including showing where the type specimens of these different groups are found, what museums they're in, who collected them, all sorts of really interesting historical information. All right. So in some of the previous examples that I've just discussed, I've talked about how genetic data has really helped to resolve complicated issues in taxonomy and classification. But as I wrap up this section of the talk, I want to dive a little deeper into how exactly genetic data is used in taxonomy and what it can and cannot tell us. First, let's consider this conceptual diagram of the speciation process, where we start with an ancestor at the top here that splits into 2 populations, and over time evolves into 2 distinct species, species 1 and species 2. When this ancestral population first splits, right around here, we do not have 2 distinct species yet. But by the time we get all the way to the bottom, the species differences are obvious. But this is a continuous process that is happening all the time. We could be catching this process at any point from the top to the bottom. And we use genetic data to understand where in this process we are. As species split, there's often gene flow back and forth these 2 populations until they finally reach a point where they're different enough that they can no longer interbreed. And this portion, this time period of the process is our gray area. All these examples that I've talked about so far fall into this gray area. Whether the things in this stage of the process are species depends on your definition of species, as well as other factors such as behavior or ecological differences. So to wrap up, I wanted to dive into some of the case studies that illustrate what this gray area looks like in a little more detail and how genetic data can tell us where we are in this continuum of speciation. In studies of hybridization, we are definitely right in the middle of that gray area. But extent of gene flow is only one component of understanding what we're looking at. The geographic distribution of that gene flow, whether first generation hybrids or later generation hybrids are more common, the timescales involved, behavior, climate, ecology, these are all important aspects to consider in determining whether or not the taxa that are hybridizing should be recognized as distinct species. Similarly, just looking at genetic divergence only tells us part of the story. In the case of the Aileron warblers, for instance, there is gene flow, but that gene flow appears to be facilitated by little assortative mating. In a hybrid zone between red-naped and red-breasted sapsuckers, which I studied for my graduate work, we also see gene flow, but the patterns of gene flow suggest that there is assortative mating. In addition to patterns of gene flow, we can look at genetic divergence between populations as a cue to where in this continuum we are. So if we look at two additional examples, we can look at common ravens and euphonias. In the case of the common raven, there is a very old split in mitochondrial DNA between birds in California and the rest of North America. But when you look at other genes and the rest of the genome for that matter, that split disappears. And it shows evidence of widespread gene flow and no contemporary isolation. So there may have been an old split, but not looking at the whole story, you lose a lot of what's going on in this case. We can contrast this with the recently split West Mexican euphonia, which shows deep divergence from scrub euphonia. But this divergence was coupled with differences in song and plumage and are consistent with differences between other euphonia species. I could really just fill an entire talk comparing and contrasting these different examples and have a lot of fun doing it, and how they fit into the speciation continuum and what genetic data can actually tell us. But what I want to leave you with tonight as I wrap this up is the importance of taking a holistic approach to taxonomy and classification, and one that incorporates many different lines of evidence as we think about different checklists and different approaches to recognizing species. Whoop. There we go.
Well, thank you very much for really a fascinating presentation. Mark, do you want to look at questions?
Absolutely.
Yeah, Brooke and Shawn, I really, really enjoyed that. I'm sorry, Shawn, I thought you had one other thing for us, but I guess I misunderstood what you had said. Sorry, I didn't mean to leave you hanging there for
Oh, no worries, no worries.
Okay.
Yeah, I had some extra information in another slide that kind of just ended up at the end there.
Okay, great. So we do have some questions. So Ken asks, is it feasible to have more detail on family membership in the systematics tab? Such inclusion would facilitate to me to reconcile a species family today from what I learned 50 years ago.
Yeah, that's a great question. And it's a need that we are thinking about addressing presently. So what appears right now in the systematics tab, there are two sort of relevant sections to help a reader understand those sort of relationships. The first is systematics history, and that section is intended to allow the reader to get some context on what this species is. Maybe you learned it as something completely different. Maybe you learned it as mule gull, and now it's short-billed gull, and you're confused. So that that history, systematics history section is intended to give that context, but also the related species section usually goes into sort of higher level relationships. But in the same token, we know we want to invest more effort and more development into our family pages. Shawn is the leader of that one.
Yeah, I certainly have a lot of ideas for expanding the family pages. Currently, as they're presented, they're kind of brief overviews of a family, to give just a brief understanding of some of the natural history highlights of a family, as well as higher level family relationships. So what other families that family is related to. But if you're interested too in looking at where a species, what family a species falls in, at the top of a Birds of the World account, we also list the genus and family that it's found in, and you can click on both of those, and it will take you to all the species in that genus or the family page. So you can also get an idea of where a species falls taxonomically with those views. But we do have plans to expand family accounts and make them a much deeper resource than they currently are. But it's a big endeavor.
Great, thank you. Let's see, John and Erica. Can you explain associative mating? I assume I heard that correctly, says John and Erica.
Yeah, so assortative mating is basically just the idea that when there is assortative mating, the same species, a species will mate with others of the same species. When there is not assortative mating, a species will mate just as frequently with a member of its own species as with a member of another species. So in the case of the yellow-rumped warbler system, a myrtle warbler in the hybrid zone is just as likely to mate with another myrtle warbler as it is with an Audubon's warbler.
So I think what Shawn just said is myrtle warblers are the problem, not Audubon's warblers.
It goes both ways.
Oh, okay.
Okay. John and Erica also asked, does Birds of the World integrate IUCN range data and use the same conservation status found there?
So we do include the IUCN conservation status on our conservation and management page. We have a less direct relationship with the range data. We show range maps via a collaboration with BirdLife. And so BirdLife's range maps are in a large part informed by that. So that information is there in accounts, I guess is the short answer.
Okay. Somewhere, I think it's in the chat, there's also a question by Lance. How does Birds of the World deal with ferruginous pygmy owls?
We're for them.
Great.
I think that pretty much answers it. There you go, Lance.
The pro owl party, as opposed to the yes, yeah. Yeah.
We do have a full species account for that, and I think it's been treated as a full species throughout its revision history.
So owl or nothing. Got it.
Hey, I just don't give a hoot for this conversation.
So Scott asks, are the entire genome sequenced to determine speciation? How does one decide how many loci needed to differ before it can be defined as a separate species?
That is a question that taxonomists and systematists constantly struggle to answer. And there is no one answer there. You can have groups that show extremely, extremely little genetic divergence. A really interesting example is the radiation of cappuccino seed eaters in South America. They basically have very extremely, extremely low genetic divergence. In most loci that you look at, they're the same, but they differ in key aspects of plumage genes, and they appear to, as far as we can tell, mate assortatively. So they don't interbreed with each other. But you have cases with similar levels of divergence in a whole suite of other groups where there's a lot of random mating and interbreeding among populations. So it really depends on the system, is really the answer. And I know that's not a very satisfying answer. I certainly wish it was easier. But yeah, in genetic data, genetic divergence alone isn't really enough to tell what's going on.
So I have a question about red poles. Now, there's an example of,
Yeah.
In terms, can you talk about that a little bit?
Yeah, so that's another really complicated situation, and I think if you asked, five ornithologists, you might get 5 different answers on how they want to treat that group. It certainly seems like a lot of the evidence suggests that they might not be valid species, but that's a case where, we're constantly learning more about them. And, it's another one of those cases where we're taking a conservative approach to taxonomy. And because people are still researching the group, we haven't wanted to make that final decision to split them in case we find out that, oh, they really are, good species, and go back and resplit them. That back and forth creates a lot of instability. And so we're kind of hoping for more research before we make our final call.
Okay, great.
And
there's
you
some things in the chat too.
Yeah.
Joshua says, in regards to assortative mating in species that have many subspecies that have more overlapping areas, would assortative mating be mediated by song as well as by appearance, and how would the hybrid offspring lean towards?
That's another. It really, I hate to sound like a broken record, but it really is a case-by-case basis. And that's what makes a lot of these things so hard, is there is no hard and fast rule when you are in that gray area of the speciation process. When you haven't, reached all the way to completely separate, it really depends on a whole bunch of factors. And yeah, some species, song is very important, but there aren't really any good predictors. I at least I can't think of any off the top of my head. I could be forgetting some recent studies that came out, but I don't think as far as we know, there really aren't any good predictors of when species are more likely to hybridize, when they're less likely to hybridize, whether there's any real contributing factors it's really, a lot of it really is a case by case basis, and it makes our jobs a lot harder. That's for sure. But it keeps us busy.
Yeah,
Yes, yes.
very
I have
important.
one practical question. You know, when I'm out doing international birding and I open my field guide, it's listed by common names, right? So how does BOW really deal with common names? Because it's a real confusion, because there's, as much as the scientific names and relationships are different, the common names are more so. So how do you handle all that mess?
So with common names, we have actually, through eBird, we show, or we're able to show and track a whole bunch of regional common names for every species. I think we're up to common names in something like 70 different languages, or regional variations of common names. So we do have a lot of information on that.
Do you get down to the group level? So it's the ones that, like, for HBW has 3 species and they're, you know,
Yeah. I don't think we have right now group level common, like a database of group level common names. But with the groups, you know, we at least have, it takes a little bit more legwork, but you know, everything is associated with the scientific name as well. So it's harder to track down, and yeah, we don't have, that would be helpful to have that database, but it's not something we currently have.
There is the nomenclature section, which gives contributors the ability to go into as much detail as necessary about common names. So we right now have a project that is working on adding indigenous names for species across the world. And so we're trying to make that section really useful in as much as possible.
Well, that's great. I'd be interested to see the indigenous names.
Lance also has in the chat, speaking of controversial splits, one that's near and dear to us in our area is, what about sagebrush versus bell sparrows?
That's actually not a system I'm very familiar with. I haven't read a lot of the literature on that particular split, so I actually don't have a good handle on what's going on there.
No.
I don't think anybody does, but
We tried, at least in the field. Great, thank you. Brittany asks, can spotted eastern or hybrid towhees be distinguished by their vocalizations?
Yeah.
I'm not. Well, we do have recordings of a whole bunch of towhees from the hybrid zone, and I haven't worked on those yet. But it is my understanding that hybrids do tend to sound somewhat intermediate between eastern and spotted towhee. The towhee hybrid zone is a mess, and it's very complicated. And I actually am working on a paper still to help sort out what's going on there. So stay tuned for the future. I hope to learn a little bit more about what's going on there. But the towhee hybrid zone is complicated.
Hey, thank you. Ted has a question. Oh boy. He says, slightly off topic about hybrid sapsucker ID. I'm curious how one would separate red-naped versus yellow-bellied parentage with a red-breasted sapsucker hybrid. Very slightly differently, which traits would present differently?
So in this case, how would you separate a red-breasted yellow-bellied sapsucker hybrid and a red-breasted red-naped sapsucker hybrid?
I think
That
that's what Ted is asking.
that's
Yes.
that's a really good question, and I really don't know if you can. And the more I've looked at sapsuckers and thought about sapsuckers, the more I'm beginning to come to the realization that an out of range hybrid that's one of these two might not be identifiable in the field. Most of my, or all of my work in sapsuckers has been on the breeding grounds. And so there hasn't been any need to think about that identification. There's currently work being done by folks in British Columbia, Libby Natola and Darren Irwin on sapsucker hybrids that includes that particular hybrid zone as well. And that might be able to shed some light on this question. But as far as I know, I don't know if there is a reliable way to separate those two hybrids.
Okay, thank you.
And Ted says thank you.
So
And
one thing, do you want to, I saw at the beginning of your slides that there was a webinar 20%. You want to say something about that?
you're muted.
Sorry about that. So there is 20% off with the code webinar 20, which I'm putting in the comments there if anybody is interested in subscribing. So yeah.
Great. Thank you very much, Brooke.
Great. We need to preserve this somehow because the chat goes away as soon as the webinar ends.
It does, but that's okay, Mark. I think we can type it down.
Yes. Thank you. Yes, we can.
Okay, actually, we had another question pop up in the chat. How does the Cornell family of databases deal with data from understudied regions? That's from Joshua. Shawn or Brooke?
I don't know if I really have a good answer for that.
Okay.
I might not be understanding the question right either.
Well, he
the Himalayan regions. How do you?
So is this like in terms of eBird data? I mean, I think yeah, I don't really have a good way of answering.
In terms of
like if is there enough data to determine what would be flagged as a rare bird in some region like in the Himalayas?
My understanding is that a lot of stuff like that, when there isn't necessarily a lot of data, it relies on regional experts to set the filter limits in eBird. So there might not be a lot of data on a certain species in eBird, but local experts know that this really isn't that rare, and so it won't be flagged. So it really relies on local experts to fill in gaps like that.
Sure.
And I should say that we do have quite an active community of contributors from kind of the Himalayan region. So from the perspective of our project, we're trying to fill in those gaps in as much as possible. So.
Okay, great.
I don't know. If there's a way to find out who the reviewers are, I'm not really that tapped into the eBird side of things.
Maybe next week when John Garrett's here, you can ask him. He might know.
Yeah. Yeah.
He'll probably say he could find out, but
I have a feeling Joshua will be back next week, so that'll be great. Well, I think we're running out of questions in our Q&A and chat, and I just wanted to thank you both, Brooke and Shawn, for a wonderful presentation tonight, especially from you, Shawn. Thank you very much for staying up so late. Straight from Ithaca, so I appreciate that. Mark?
My pleasure.
Yep. Yeah, thank you so much. It was fantastic, and
Yeah.
we'll be putting it up on the web for people to look at later.
And I just want to remind everyone that next Tuesday night, first of all, next Thursday night, 2 nights from tonight at 7 o'clock, we will have our final get-together meeting for the Trial LA Bird Atlas, Winter Bird Atlas. Please register beforehand and join us. It will be recorded, but there's only a few days left for the segment that we'll be counting, which ends on February 15th. So time is running out. Time is imperative, is important. Be there. Okay, next Tuesday we'll have John Garrett here, who will talk about eBird and answer Joshua's questions. And I think that's it. Once again, thank you very much, Shawn and Brooke, for being
Thank you
with us
very much.
tonight.
Thank you, everyone.
Thank
Really
you so much for
informative.
having us. It's been a lot of fun.
Such
It-
a huge project, really impressive.
Incredible.
Yeah.
And so useful.
Yeah, thank you for all you do.
Yes.
Absolutely. And we're getting tons and tons of thank yous in the chat. So I'll incorporate them all together and say thank you very much. Hopefully we'll see you on a future webinar, and we'll see you all next Thursday and Tuesday. Take care, everyone.
Good night, everyone.
Good night.